Hi, I'm Megan Steinmetz, and this was my dissection of a sheep eyeball. So I first looked at the optic nerve, which sends impulses to the brain from the retina. The sclera, which is the white of the eyeball, and the cornea, which forms the front of the eye. I made my first cut, which is what you're looking at now. Would have been that. Made my first cut posterior to the middle of the eyeball. This was not the consistency that I expected at all for the eyeball. It was much more dense and thick, and I expected it to be a lot thinner because our eyes are such a sensitive part of our body. Once I was able to separate the eyeball, the viscerous humor drained out, and this usually helps maintain the shape of the eye, which is something I could kind of feel before, just kind of keeping the formation of the eye together a little bit. We can then see the cavity between the retina and the back of the lens called the viscerous body. The lens directly posterior to the iris and the pupil help focus light and image of the retina. The hyaloid fossa is a depression on the anterior surface of the viscerous body. I then cut out the front half around the cornea, as you can see right here. So I just cut it around right in there. And I kind of cut that in half too a little bit, just to kind of see how thick it was. And this also really surprised me because I pictured this to be a very thin membrane. I then looked further into the iris, which contracts pupil size, and I did not remove this because due to the size of it, I wanted to well preserve it and reference it. So if I focus more on the back part of the eye, you have the retina, which connects all the way into the optic nerve. And here, the blind spot, which is caused by an absence of photoreceptors. And the photoreceptors are commonly the rods and the cones, and these are responsible for our color vision, something very important. On this side, we can also see the optic nerve and the fibers of the nerve. If I move the retina and look inferior of the back of the eyeball, I can see this beautiful shining, tissue, which is the coracoid, followed by that pretty turquoise color portion, which is the tapetum lucidum, which helps a person's night vision. This was a very interesting lab. It was a little bit harder, I found, just with the size of the eyeball and not expecting the thick, dense tissue, as I envisioned it a little bit thicker. But it was a very interesting lab, I thought. Thank you.